quickly revise what we did last week because I'm going step by step and keeping the flow until we make recombinant organism. After that, rest of the information and other things we can discuss later. That's very easy to understand. If we add many things in between, we're going to miss the flow. Okay. First thing I mentioned, recombinant DNA transfer DNA from one organism to another organism. In that case, it could be a organism A to C, or we can combine A DNA piece of B DNA together, and A and B we can transfer to C. So this is called genetically modified organism or transgenic or recombinant DNA. Now the aim of the lesson is to learn whole process step by step until we make organism C. And that case again it become a bit complicated because we can transfer to animal, plant, fungi, and bacteria, and even if we want we can transfer to protists, but mostly bacteria we're going to use here for the understanding. But uh, certain cases we can also learn plant and fungi like yeast, how we can transfer gene to plant and fungi. Animal, we are not talking within this level. A resource book does not cover the animal transfer gene to animal, so we are not going to learn that thing. But we'll mostly focus on bacteria. Okay, here it gives again overview. We can transfer to bacteria, to yeast, plant, animal, and also to a we can use virus. We use virus not to directly transfer genes, but we transfer genes to virus, and then we use this virus for other purposes. And main three areas: agriculture, research, and medicine. Okay, we're going to learn the whole procedure in several steps. ICMITIC. So these are the main steps we're going to cover in recombinant gene technology. First, we learn isolation of DNA. I is for isolation of DNA and identification of DNA. So what we learn there, we can use a mechanical method or chemical method, and we can disrupt the cells and we can take out the DNA. During this procedure, we have to add certain things. This is an enzyme. We can lyse the cell membrane and we can take the DNA out of the cells. One of the important steps we should keep in mind always, we have to add DNase inhibitors. DNase are the enzymes which can break DNA, which can hydrolyze the bonds of the DNA, which present in the cells in lysosome, or which could present in even a surface, because when the bacteria uh, contaminate the surface, if the bacteria die, this DNAs can release to the surface. So there is a chance of contamination while we are working with uh, the DNA isolation procedure. So we have to add inhibitors for DNAs. And then once we get the DNA out, we have to remove the protein like histones to purify as a step of purification process. So we can use certain detergents like SDS, phenol, and proteolytic enzymes. Then after that, we can precipitate DNA, and we can wash the DNA several times, get the pure DNA. Precipitation done by cold alcohol or chloroform like chemicals, because first we add the salt, sodium ion, neutralize the charge of DNA molecule, because DNA is negatively charged, then there's no charge. When there's no charge, no polarity, we can use alcohol-like solution to precipitate it because it's not soluble. And then this is something I should make a small note. I just use this NRB. NRB is not in resource book. So not in resource book, that means you don't want to really make effort to remember these things, but just not bad for some kind of additional knowledge, because more than isolation of DNA nowadays, the method is DNA isolate from messenger RNA. That's a common method nowadays. So just we have to update a little bit with the common methods, still keeping a small remark, not in a resource book. That means there is no chance of asking in case something related to this, we can still make it, because when we isolate mRNA, reverse transcriptase, enzyme can make cDNA or complementary DNA. Then DNA polymerase can make it double strand. So then this method is much convenient and much better, because messenger RNA do not have uh, introns. So we get the correct sequence of the gene. That's why people try to use this method, and not like a DNA. DNA has one copy of a gene in a cell. Cell produce many copies of mRNA, so we can also get more copy of the genes if we do this method. Because of these two advantages, this become one of the most common method nowadays, rather than isolation of DNA. Okay, then we have to identify the correct gene of the complete DNA molecules. When you get the DNA, it's a whole DNA molecule in cell. So we need to get the correct sequence. For that, we use restriction endonucleases. That's enzymes, restriction endonucleases, and they can identify restriction site, because along the DNA molecule, there are sequence, and the sequence are called restriction sites. Restriction site. The restriction site usually there is a different length along the DNA. So here you see distance is shorter, this distance is longer. So the distribution is random, and as a result, we get different fragments, because if we use a restriction enzyme, it identifies the sequence, not the shape, because DNA molecule shape is same from one end to another end. There is no shape for enzymes. It identifies the sequence. As a result, it can hydrolyze the phosphodiester bond like this. And this is called restriction site. So again, we get fragments of DNA after that. Here you see resulted several fragments. Sometimes we use one restriction enzyme. Sometimes we use several restriction enzymes together to cut the DNA into small fragments. So one of the fragments we can use as our gene. One of the fragments we can use as our gene. Now the problem is how to identify that I have a correct fragments containing the gene. So we need to confirm that I have a correct fragments. For that, we do agarose gel electrophoresis. Agarose gel electrophoresis separate DNA molecule based on the size. Based on the size. Because different fragments have different size. So this is a separation of DNA molecule. We can also use this technique for protein separation. And we place the mixture of DNA in the electrical field DNA molecule move from negative side to positive side. Then larger molecule can move slower, smaller molecule can move faster. As a result, we can see different bands in the DNA molecule, the gel. So it can run through the gel, and then we can see like this different bands. One of the bands is our gene. So how do we identify that the band is our gene? So we also use a marker fragments, that mean known weight of the fragments. Usually the weight is given as a base pairs. So commercially available this DNA marker, it's also called DNA ladder, reference band. So we run this one together with our samples. As a result, when we roughly estimate my gene base pairs is let's say 700. So if it is 700, then I can take these two bands, because this is a one lane, this is a second lane, for example, and then I can isolate. I can guess, okay, it is in front of 700 base pairs. This may be my gene. And here in a real picture, you can see like this. We use this UV light with the marker ethylene bromide is a marker used. Ethylene bromide give this kind of a visible band um, when we expose to UV light. Because if this is the DNA, ethylene bromide bind to it, and we can track the band location. Right, I think we discussed the last class until that. Before we proceed to the next section, I found a few questions. We can still do these questions. I mean, you can try first, right? So I just leave to you whether you can identify the correct answer for 31, question number 31. Genetically modified organism is different from other members of the same species because
second answer.
Yeah, I think it's a quite easy one. We learn genetically modified organism contain foreign DNA. So a gene or genes, as I told you before, it can go to A, B organism and go to C. We can go transfer A to C, or we can combine A and B together to C. So gene or genes from another organism. That's one gene here and one gene here. Then we can place it here. Next one, we also learn genetic counseling. So I mentioned everything about genetic counseling, qualification, what kind of person and what is he doing, and how it works in the hospitals.
Second answer. No, incorrect. Second answer. Third answer.
Third one. That's the second one, third one. Yeah, sorry, just the wrong one. Erase. Yeah, third one.
Yes.
Yeah, third one is correct because one of the most important concept in medicine is it's called patient autonomy. Patient autonomy. That means we have treatment, we have different remedies, we have different methods when there is a disease, when there is a problem. The goal, the main objective of a doctor is to diagnose what is the correct problem of a patient. Once doctor diagnose correctly the problem, then doctor can offer to the patient, you have these options. So he can explain these options to the patient. When gave this option to the patient has several, patient can make few decisions. Decision number one, patient can reject. Patient can reject everything. No, I don't want any of the options. I live with my disease. It is my disease. So that is a one option, because let's say there's a kidney failure. Then at the end of last stage of kidney failure, then doctor can advise, now you have no option. You need transplantation, kidney transplantation. And the patient can say, no, I live with this, my life, that's okay. Even the family members cannot force him go for a remedy. So it's a patient autonomy. The patient autonomy is very important in medicine, that patient can decide herself or himself what he's going to do. In that case, he can't advise to abort. He can only give options. Option is, okay, the fetus has genetic disease. This genetic disease is a mild disease. It could be a not life-threatening. He can live as a normal person at least 50, 60 years. Let's say Down syndrome. So they diagnose Down syndrome and they say, okay, he can live. But he can't say, okay, if you don't like the baby with the Down syndrome, I really advise you to abort the fetus. He cannot say. He can say, the baby will born with the Down syndrome. And if the patient asks, what can we do? You have options, several options. One is you can decide if you want to abort the baby, you can decide. And some countries, this is again not that easy. Medical abortion is accepted, and again medical abortion is again not that easy, depend on the situation. But he cannot advise to abort the fetus if one of the parent is carried of a genetic disorder, because one of the parent is carried of genetic disorder. That's also not 100% sure that the baby will get this genetic disorder, because we know both recessive alleles should combine to make the phenotype in certain cases, and a risk is less, but still advising him to abort. That's wrong. Even they detect some kind of fatal genetic disorder, they cannot insist on parents to abort the fetus, but they can give option. This is one option you have. If you would like, you can go for it, because this condition is fatal. If baby born with this genetic disorder can live only for few months or few years. So in that case, you can decide, it is your decision whether you're going for abortion or not. But other case, if the baby can live with the genetic disorders as normal life, that's again, it's very difficult situation. So ethics are coming to this place. Ethics is not the law, it is a personal and belief of the society, moral issues, so religious issues. So things are not that easy, even we have a solution in biology. Okay, that's second answer. You both correctly identified. Very good. Right. Now, continuation of the lesson. How do we know the band we get is exactly the gene I wanted to transfer? I wanted to make recombinant DNA of another organism. In that case, we have to rely on the sequence, because sequence of the gene is a very strong evidence. This doesn't change. So We now go for sequence to look the sequence. So we can go for sequence. Now in that case, the method is called DNA probe and the southern blotting. So it is called DNA probes and southern blotting or hybridization. DNA probe and hybridization or southern blotting. Okay, let's learn what is DNA probe and hybridization and southern blotting and how do we do this. The person who actually discovered this method, his name is Southern. So then we also call this method southern blotting. Right. Okay. Now this gel is very fragile. It's very difficult to work with the gels because even handling gels sometimes it can break. We have to very carefully handle this gel. Uh, if a person is new, there's a higher chance of breaking the gel. But once a person uh, they know it how to do it, then they can handle it. For example, most of the time, it doesn't matter what is your background, MBBS or science, agriculture or BSc. People have to go for higher education. When they go for higher education, all the people are same when they are doing their research work to do get the master's, PhD side degree. They should do themselves most of the time this kind of a work. They learn it and they do in the lab. All these techniques, they do it. Right? Because sometimes MBBS degrees or medical doctors, they have to do research until they become qualified to go for a next consultant level. So some countries, they have to do a research and they have to find something and they have to publish it. So that is a qualification to go higher level in this career. Now this, how the gel looks like. It's very thin, not maybe half a centimeter thickness, and it's very fragile. Now first aim is we have to transfer bands to a suitable flat form. So gel is very fragile. We can't use the gel anymore. All the gels, all the bands in the gel, we have to transfer to another material. In that case, we select a paper. The paper is not exactly same as the paper printed our textbook like things. It is a mixture of nylon and cellulose. Nylon and cellulose. So it's more toward the nylon because nylon is it's like a little bit like a plastic sheet, but when it mixes with cellulose, it gives a good texture. It's a nylon and cellulose paper. It is called nitrocellulose paper. So we use this nitrocellulose paper, and other advantage of nitrocellulose paper is it's a positively charged. Surface is positively charged. So why it is advantage now? Because DNA is a negatively charged. So then the DNA can strongly bind to the surface. This can strongly bind to the surface, being nitrocellulose is positively charged. So this is the method now we're going to use. Now let's see how can we do this. What are the steps of transferring bands to the nitrocellulose membrane? First of all, we need to denature the DNA molecule. Denature in the sense we're going to separate the DNA into single strand. We're going to separate DNA into single strand. We will see why it is important because we're going to hybridize with the known sequence. So for that, hydrogen bond should be broken. After that, what we get a single band like this C, T, A, G. So now it is attached to the nitrocellulose membrane. This is the expectation at the end of this hybridization process. So we need to get a single strand DNA firmly attached to nitrocellulose membrane. Okay, let's see how we can do this. This is a nitrocellulose membrane, just a piece of paper. So it has a small box, and there are 100 around papers we can see which fit into the gel. Now, first step in this case, we have to separate the double strand into single strand. For that, we're going to use alkaline medium. So if we place the gel in an alkaline medium, then these two strands are separated. When the two strands are separated, that means hydrogen bonds breaks down. These two strands separate, and in alkaline medium, they try to stay as a single strand. Then after that, we are going to the next level. That is the gel, and that's a paper. So we're going to transfer this single strand DNA to this nitrocellulose paper. For that case, we're going to use a method, a simple method, the capillary action of water. That means if we place the gel like this, and top of that, if we place the paper, and then we place this in a buffer solution, not that so if you place in a buffer solution like this, then the buffer solution trying to move from high water potential to low water potentials. Try to move from high water potential to low water potential. Then the movement is this direction. So when capillary action, the water molecules are moving, they can drag this isolated DNA of the gel to the paper. So this is the principle of this method. In the capillary actions from high water potential to low water potentials, molecules are moving upward. When the molecules are moving, DNA molecules can also.
So your voice is a bit not clear, Sasangle. Your voice is a bit not clear, Sasangle.
Give me 1 second. Okay. Is it clear now?
Now it's clear, sir.
Okay. Right. This is the gel. This one is nitrocellulose membrane. And here we're going to place papers. We call tissues or paper towels. And here also we keep tissues or paper towels. gel, paper, I just separate paper, I just nitrocellulose. That's a nitrocellulose membrane, and this is a normal filter papers. And then top of that, we can place a weight. And then we can place this in a buffer solution. So setup is like this generally. When we keep the weight on the top, it firmly press all these layers and allow water molecule, the buffer solution, to pass from this direction. So these papers on the bottom and the top are attractive surface for water molecules. Then from high water potential to low water potential, the water is moving, and when the water is moving by capillary action, the DNA molecule in the gel can also move and attach to the nitrocellulose membrane. Because nitrocellulose membrane is positively charged, DNA is negatively charged. Usually, we leave this overnight. We leave this overnight, and when you come next day morning, all the DNA molecules in the gel transfer to the nitrocellulose membrane. Here, I'm going to show you the same thing in a most correct and nice way. So here, transfer buffer, we can keep a small supportive platform, and then on the platform, we can keep this filter paper. See here, this is a filter paper. And then gel, nitrocellulose membrane, filter paper, again more paper towels, and the weight. So when we keep this, we don't see space like this. When they keep the weight, everything press. So it provides a good pathway. The buffer solution can move upward. So this paper towel, big one, serve as a reservoir. The more buffer is going, so this buffer soak the upper paper towel. It's not that complicated system, but anyway, these methods are a little bit outdated. Now we have a many advanced method, but still people are using these methods. So it's good to learn these methods, because these methods provide us the background, the real background, how really things are working. Fine. So I will again show you same thing in this animation. It's good to remember when we see this kind of a thing, rather reading and understanding and remember, visuals give us more fast way of learning. Okay, Southern Blotting Station. I just skip certain things. We go directly to the Southern Blotting. So here, first we have to do gel electrophoresis. I think we have seen gel electrophoresis. We learn gel electrophoresis. So we go to the hybridization and probe method. Okay, Southern Blotting. Transfer buffer, and we keep the support first. And then we place a paper towel and soak the paper towel with the buffer. Then it contact directly the buffer, and we keep a set of papers again. Again, we soak. Once we soak, it communicate or contact with the next level. And then we take the gel after electrophoresis, gel electrophoresis, and place on the paper towels. Then we take nitrocellulose membrane. So we place nitrocellulose membrane correct surface, and then we keep next set of papers, filter papers on nitrocellulose membrane. And this plastic wrap prevent evaporation of the buffer solution. And then again we keep paper towel, and then the glass plate and the weight. So it will completely press all the layers now and make good communication with the buffer solution. Now, due to capillary action, buffer solution is moving. The single-stranded DNA molecule move upward, and then it just attach firmly to the nitrocellulose membrane. Then we take nitrocellulose membrane, and we can wash nitrocellulose membrane. Nothing will happen because DNA firmly attached to this one. And after that, we can still visualize whether transfer is happened correctly by checking with the UV light. We can see the bands from this UV light, right? Okay. So we confirm the transfer is perfect. Nothing happened. Nothing wrong happened. Transfer is perfect. We see the picture bands similar to the gel once we look on the UV light. Okay. So we're going to the next level. Next level is DNA probe. How we can add DNA probe, and what is the last step of this hybridization method. Okay, let's try to see it again.